Hello everyone and welcome to our module on glucose. Let's start by talking about carbs or carbohydrates. So the word carbohydrate literally means a watered carbon, and that is because most carbohydrates have a chemical formula of some number of carbon molecules and then some number of H2O molecules. So for example, let's look at glucose. Glucose has a molecular formula C6H12O6. So you could think of this as 6 carbon molecules and 6 water molecules. The simplest carbohydrates are called monosaccharides, and there are 3 of them that are important for biochemistry: glucose, fructose, and galactose. All 3 of these monosaccharides have the same chemical formula, C6H12O6. What's different from one monosaccharide to the other is the spatial orientation of the molecules around each of the 6 carbons. So glucose, fructose, and galactose are all isomers of one another. They have the same chemical structure but different spatial orientation. Disaccharides are carbohydrates that contain 2 monosaccharides linked together, and disaccharides are broken down into their monosaccharide constituents in the GI tract. It's the monosaccharides that get absorbed into the blood. The 2 most important disaccharides for you to know are lactose, which is found in milk. Lactose consists of a galactose and a glucose molecule linked together. I've shown you a picture of that at the bottom of the screen. The enzyme lactase breaks this down, and you may know that some people lack this enzyme and they are called lactose intolerant. The other important disaccharide for you to know is sucrose, which is what's found in table sugar. It's the kind of sugar you put in your coffee at Starbucks, and it contains fructose, and the enzyme sucrase in the GI tract breaks this down into the monosaccharides. Complex carbohydrates are polysaccharides, so these are polymers of monosaccharides. There are lots and lots of monosaccharides linked together. You may have heard of starch. Starch is a plant polysaccharide that consists of glucose polymers. Glycogen is an animal polysaccharide. It is also glucose polymers, but these are oriented such that the human body can break them down into glucose when they need energy. And then you may have heard of cellulose. This is a plant polysaccharide of glucose molecules, but it has different bonds from starch and it cannot be broken down by animals. So when we say that people need to have fiber in their diet, we're often talking about cellulose because it's not broken down in the human GI tract. It adds bulk to our stool and improves bowel function. And for many elderly patients with constipation, you will recommend fiber in their diet, and what you are recommending is these cellulose molecules that cannot be broken down in the GI tract. So just to reiterate what I said before, all complex carbohydrates or disaccharides are broken down into either glucose, fructose, or galactose, and these are the energy sources for the body. We'll talk about fructose and galactose in another module, but glucose is far and away the most important carbohydrate to the body, and that's because it can be used for so many different things. And we're going to have separate modules on all the ways the body uses glucose. So we will talk about using glucose for anaerobic metabolism in the glycolysis module. We'll talk about how it can enter the tricarboxylic acid cycle. It can also be directed to the HMP shunt, which is how we get the ribonucleic acids we need for DNA and RNA. It's also how we get a very important molecule called NADPH. Glucose can be used to make fatty acids, and it can also be converted into glycogen. The different tissues and organs of the body use glucose differently. The liver has some of the most varied use of glucose. It can use it for the TCA cycle to generate ATP. It can also store glucose as glycogen. The brain makes constant use of glucose for the tricarboxylic acid cycle to generate ATP. The brain has very little glycogen storage, and therefore the rest of the body will often work very hard to supply the brain with a constant source of glucose. Muscle and heart tissue use glucose for the tricarboxylic acid cycle to generate ATP. The transport of glucose into these cells is heavily influenced by insulin. So when there's lots of insulin around, they take up more glucose, and they often store glucose as glycogen for later use when there is not glucose readily available. Red blood cells are unique in that they don't have mitochondria. This means they only use glucose for anaerobic metabolism to make ATP. This means they are constantly generating lactate from anaerobic metabolism, and that lactate goes back to the liver and gets converted back into glucose. Red blood cells also use some glucose for the HMP shunt to generate NADPH. Finally, adipose tissue mostly converts glucose to fatty acids, which should be obvious, and like muscle, the uptake of glucose by adipose tissue is heavily influenced by insulin. There are 2 general ways by which glucose enters cells in the body for metabolism. The way that the vast majority of cells take up glucose is through sodium-independent entry. This is a type of entry that does not require sodium co-transport. I've drawn a picture of it at the bottom of the screen. There's a high level of glucose outside the cell, a low level inside, and there is a transporter that pulls in glucose down its concentration gradient. 14 different transporters like this have been described, and they're all named GLUT1 to GLUT14 depending on where they are found, and it varies by tissue. So for example, GLUT1 is found in red blood cells, GLUT14 is found in other cells. The second type of glucose entry into cell, which is less common, is called sodium-dependent entry. This is used when glucose needs to be absorbed from a low to a high concentration. There are two important places where this takes place. The first is in the intestinal epithelium, and the second is in the renal tubules. Here's a depiction of how glucose is absorbed by intestinal epithelial cells. So in the GI lumen, you have a relatively low concentration of glucose compared to inside the cells where it is relatively high. Therefore, in order to absorb glucose, the cell will couple that absorption to sodium. There's a transporter called SGL1, which will pull in a glucose molecule along with two molecules of sodium. Inside the cell, the concentration of glucose is relatively high compared to the interstitium or the blood where it's relatively low. So therefore, one of those GLUT transporters that does not require sodium can work on the other side of the cell and allow glucose to leave the cell going down its concentration gradient like I've shown here. The sodium is then pumped out using ATP to create a high concentration of sodium outside the cell, which can then be used to drive more uptake of glucose. This is a picture of a proximal tubule cell. I also show this picture in some of the renal modules. So the concentration of glucose in the lumen of the nephron is relatively low, and it's relatively high inside the proximal tubule cells. So therefore, glucose uptake is coupled to sodium in order to pull it out of the urine. Once it's inside the cell, the concentration gets high relative to the interstitium of blood, so the glucose can leave in a sodium-independent manner out into the interstitium of blood. And in order to keep this whole system working, you need ATP pumping sodium out of the cell. Finally, let me finish by mentioning a few specifics about some of the GLUT transporters. Remember, these are the glucose transporters that do not require sodium co-transport. So GLUT1 is an insulin-independent transporter, so it will take up glucose when the glucose concentration is high, and it will take up less when the glucose concentration is low, but it's not influenced by the hormone insulin. And you can find this in the brain and in red blood cells. They basically take up glucose whenever they have the opportunity. GLUT4 is an insulin-dependent transporter, so you will find this in fat tissue and skeletal muscle, and these types of tissues and organs want to take up glucose only when insulin levels are high and then store it as either fat or as glycogen. The GLUT2 transporter is also insulin independent, but it is bidirectional, and this means that it can transport glucose out of cells for gluconeogenesis and for other reasons. So in the liver and kidney, which are the two organs that can synthesize glucose for the body via gluconeogenesis, they want this bidirectional transporter so they can both take up glucose and also put it back out into the bloodstream when the body needs it. You can find the GLUT2 transporter in the intestine. It carries glucose out of epithelial cells to the portal vein. Here's the slide I showed you a minute ago, and here's the GLUT2 transporter right here, letting glucose go out of the cell and into the interstitium or blood. And then finally, you can also find GLUT2 transporters in the pancreas. And that concludes our module on glucose.